catalysts, and enzymes.
What exactly is a catalyst and how does it work? A catalyst are substances that speed up a chemical reaction to increase reaction rate without getting consumed during the course of the reaction. We actually use catalysts to speed up the rate of a reaction, and because they're not consumed during the process, we can actually use them over and over again. And for a chemical reaction to occur, one of the main conditions is that the reactant particles must have a significant amount of energy to break their existing bonds. This minimum energy is actually called the activation energy, and so the catalyst provides an alternative route for a reaction to occur. This alternative route has a low activation energy, as we could see this on the graph. There are many types of catalysts. There are catalysts in the same phase that are called homogeneous catalysts, while those in a different phase are called heterogeneous catalysts, and we will later be talking about this.
Okay, so first we have the homogeneous catalyst. So homogeneous, basically this catalyst means that it's in the same phase as the reactant. So they're typically found in liquid or gas form. An example is the depletion of atmospheric ozone due to chlorofluorocarbon or CFC. So first CFCs are broken down and chlorine atoms are released. So initially the chlorine is removed in the first reaction over here, but it's regenerated in the second equation. And so the net result is the depletion of ozone and oxygen. The chlorine atom is not consumed in the overall reaction. It just provides a different pathway for the overall reaction to happen faster. And so the chlorine is the catalyst, and the ozone and oxygen are the reactants. So they're all in gaseous phase, thus it's a homogeneous. Okay, so next is the heterogeneous catalyst. Heterogeneous, meaning that this catalyst is in a different phase from the reactant. So these are typically made of solid metals and metal oxides. An example is the catalytic converter found in cars. So this is a catalytic converter, and it produces carbon monoxide, nitrogen monoxide, and hydrocarbons from the combustion in the car exhaust. So here, the reduction of nitrogen monoxide is catalyzed by platinum and rhodium, while platinum and palladium catalyzes the oxidation of carbon monoxide and hydrocarbons. So as a result, our cars emit less toxic gases at a faster rate thanks to these catalysts.
What exactly are enzymes? Enzymes are biological catalysts. An enzyme is a type of protein within a cell that can help speed up a very particular chemical reaction. And so the way the enzyme works is by binding a reactive molecule and holding them in such a way that the chemical bond is breaking and or the bond is forming, causing the reaction to just happen a lot faster. And so with the catalyst, the activation energy is a lot lower than without. This lowering is important because the enzyme has to transfer reactants into a product. There are 7 types of reactions that the enzymes are catalyzed in, all using the suffix of "ase". We will later go into more depth of each one of them. Active site. The active site is where the enzymes will grab onto a reactive molecule. These molecules are the enzyme substrates, the part of the enzyme where the substrate binds. A substrate enters the active site of the enzyme, and so this form is called the enzyme substrate complex.
It was mentioned earlier that there are 7 classifications of enzymes, so let's go to each of them in detail. The first one is called the oxidoreductase. So this enzyme is involved in redox reactions. An example of this enzyme is the luciferase, which functions in the bioluminescence of fireflies. Next is the transferase. So this enzyme assists in the transfer of functional groups from one molecule to another. An example of this is kinase. So this adds a phosphate group to other molecules. Next is the hydrolase. So from the word hydrolysis, basically it's a process that breaks down bonds by addition of water. So hydrolase basically accelerates hydrolysis. An example of this enzyme is lipase, which breaks down triglycerides. Next is the lyase. So lyase enzyme breaks down bonds by means other than hydrolysis or redox. An example is pectate lyase. It's found in plants and it's involved in the degradation of the cell wall. Next is the isomerase. So from the word isomer, isomers are compounds that have the same chemical formula but have different molecular structures. So isomerase catalyzes the rearrangement of these atoms in the molecular structure. Okay, so example is xylose isomerase, also known as glucose isomerase, is pictured here. It's used to convert glucose to fructose. Next is ligase. So this one joins two molecules to form a single compound. Example is DNA ligase. As seen here, it repairs double strand damage in the DNA. Last but not the least is the translocase. So this promotes the movement of ions or molecules across a membrane or separation from the membrane. Example is the ADP-ATP translocase. So here, that enzyme transports ADP into the membrane and ATP out of the membrane.
Factors that affect enzyme. The first one is temperature. Since enzymes are macromolecule classes of protein, the rate of reaction increases as the temperature increases. So when the temperature is low, it's going to have less kinetic energy and it's going to result in inactive enzyme. And then as the temperature increases and past a certain point, your enzyme changes shape and results in denaturing. And that's why I wrote right here that the rate of inactivity is highest within its optimal temperature. The second factor is pH. pH can also affect enzyme because when you change the pH of a solution the enzyme is working in, your enzyme becomes misshapen or denature. So let's say that this enzyme right here is put into a new solution, could be acidic or basic, but if the pH is different enough, it changes the shape of the enzyme, causing not to bind to its proper substrate. So yeah, the graph right here shows you a vertical line in the middle, indicating where the enzyme works best. And then the last factor is the substrate concentration. Same idea right here, that they have a proportional relationship but to a certain extent. So here at a low concentration, many enzymes are just waiting around for a substrate. And at a high concentration, all the enzymes are hard at work and the extra substrates are waiting around for free enzyme. But then when it's close to the maximum speed of the enzyme, the increase in concentration has a negligible effect on the rate of reaction. The last thing I want to cover is the induced fit mechanism. Scientists used to think that these enzyme binding work like a lock and key, but actually it doesn't work like two puzzle pieces fitting together. Lock and key is the basic idea that the substrate fit exactly with the active site of the enzyme, but in the induced fit mechanism, it explains how the substrate enters the active site, changing the shape of the enzyme slightly so that it fits even tighter with the substrate. So, in conclusion, this one, the induced fit mechanism, is newer and is a better represented model.
Now we're going to talk about the differences between catalysts and enzymes. The differences between catalysts and enzymes are their structure, because while all enzymes can be considered biological catalysts, not all catalysts can be considered enzymes. Catalysts are simple inorganic molecules that are not specific to any reaction, which means they can increase the rates of many reactions. Enzymes, on the other hand, are exclusively organic and complex proteins that are highly specific, which is based on their active site. Proteins consist of amino acid chains that are folded into arrangements based on the amino acid chain sequence, which is also known as its primary structure. This primary structure essentially just gives the direction to the enzyme or a protein for how it's going to be folded, like said, or for its overall 3D structure, which gives the shape of the active site. Another difference between catalysts and enzymes are the conditions for which reactions are able to occur. For catalyst reactions, they can actually still require additional energy inputs, such as a higher temperature, to reach the necessary activation energy. Enzymes are inorganic life forms which already maintain a stable and ideal condition for these reactions to occur. So changes in that environment can actually denature the enzyme, as discussed previously.
Okay, so now that you understand the difference between catalysts and enzyme, let's look at their relationship to reaction profiles. Okay, so first of all, it is important to understand that catalysts, enzymes, allow the reactions to initiate or occur at a lower energy. That is the most important thing that we have to understand here. We also need to understand that while the initiation or energy, we are not completely using up these enzymes in the reaction itself. We are just start using it to start or let the reaction occur at a lower energy. Or using these enzymes specifically, we help to lower the free activation energy in a reaction, meaning the transition state where which is a state after the reactants and right before the products where all the bond breaking and things occurs. So these enzymes specifically help us achieve that state through much lower energy. Okay, so how does enzymes do this? They use catalytic strategies to allow these reactions to lower energy. So these strategies include acid-base, covalent catalysis, electrostatic catalysis, and proximity orientation effects. The first one, which is acid-base, it actually helps through transfer in the reaction. And then we have the covalent transfer. Here the enzymes actually helps by covalently reacting molecules to help reaction through lower energy. And then we have the electrostatic strategy where use charged molecules and metal ions to stabilize certain charges in the reaction to let it occur with a low energy. And then we have the proximity orientation, which help increase the collision of the reactants in order for the reaction to carry on with lower energy. And here you can see that our reaction without the enzyme requires a lot of energy, while it does not require as much energy with enzymes being present.
We are now moving on to the quiz activity portion of this presentation. Once a question is read, please pause the video and write down your answers. Given the reaction, determine the catalyst and draw its reaction profile, although it doesn't need to be super specific. Glucose plus ATP plus hexokinase reacts to form glucose 6-phosphate plus ADP plus hexokinase. True or false? All catalyst reactions have a decreased change in enthalpy, also known as delta H, when compared to the same reaction without a catalyst. Which of the following is not true for catalysts/enzymes? A. Enzyme reactants are referred to as substrates. B. Catalyzed reactions are temperature dependent. C. Lyases break down molecules by hydrolysis. Or D. Increasing the temperature of a reaction will always increase its rate. An enzyme will be used in the synthesis of ATP from ADP and a phosphate group.